The topic I chose for this paper was genome editing. The reason I chose this is because in recent years there have been many revolutionary strides that have made genetic engineering through gene editing and gene therapy extremely important. It is happening at a quicker rate than ever, and I believe that it is important that we understand why. To start, I'll be talking about what is genetic engineering. Genetic engineering, also called genetic modification or genetic manipulation, is the modification and manipulation of an organism's genes using technology. This can be done by deleting regions of DNA and adding new segments of DNA. This is primarily subclassed into gene therapy and gene editing, having a key distinction being their purpose. Genetic engineering aims to modify and enhance the genes to better capabilities than what is normally intended. As times pass, we have been able to have success with genome editing as knowledge grows and more safety data becomes available to help develop better approaches to this process. The development of more specific and efficient gene transfer vectors that are now being used in trials. This includes that containing of active substances which consist of processes that uses pieces of broken off DNA recombined with these pieces into new alleles through nucleic acids in hopes of replacing, regulating, repairing, adding, or repairing the deleted genetic sequences. One of the processes of genome editing is gene therapy. Gene therapy allows us to alter the genes to correct genetic defects. It helps to prevent or create cures for genetic diseases, and a process that this includes is somatic editing. Somatic editing edits materials that are inserted into the targeted cells but don't cause changes that are passed down to later generations. The other process is genetic engineering. Genetic engineering modifies genes to enhance the capabilities of organisms beyond the normal level. This is done through germline process of editing. This is different from somatic cells, as somatic cells repair one's body, while germline cells are done through reproductive cells, as they can be passed on to later generations. For the history of genome editing, in 1987 the identification of repeats in the bacterial genome were found, allowing us to have an understanding of genetics. In 2002, CRISPR was termed. CRISPR is the basis of the immunity in bacteria and archaea that helps the use of enzymes to help bind and create double-stranded breaks. In 2012, CRISPR was found as a programmable endonuclease. In 2013, eukaryotic gene editing using CRISPR-9 and Cas9 was possible. In 2013, this CRISPR and Cas9 usage was found to correct the genetic diseases within mice. In 2014, the same process was used in primate embryos. And in 2015, that same process was used in human embryos, as well as major applications were used with somatic human genome editing, with vivo editing. This is done within a human tests and procedures to address HIV, sickle cell disease, and more, stretching across a wide range of diseases, including genetic diseases and cancer. In 2018, many clinical trials have begun with this process, and in 2019, we began engineering prime editors. These prime editors can make useful targeted insertions, deletions, and other changes to DNA, making it much more useful and much easier. For why should we use genome editing? Genome editing can be applied to genetic diseases/defects, cancer treatments and cures, and other uncurable known diseases. Defects can also be included as ADHD, autism, and other predisposed defects within an embryo that can possibly be changed. The picture to the left shows the CRISPR and the Cas9 being used and how it is used. An example of an in embryo is the utero gene therapy. This treatment is suitable when it's possible for a pregnant woman who already has a fetus with a hereditary disorder that is diagnosed and does not want to go through an abortion. This can be possible to make the corrections to the genetic defects during development, allowing the fetus to possibly avoid the early onset of genetic defects, allowing for better brain functions and also allowing for permanent correction of possible organ functions and avoiding possible immune response complications. This brings us to balancing the ethics. In recent years, we have seen a drastic advancement and a new frontier being created from this exponentially growing field. With this, we see a large acceptance of genetic editing as it can be very useful. But at the same time, there is causes for concern of safety, as well as a great worry of using this for more than just survival or improvement of health of an individual, but instead for the exploitation so that one can improve the aesthetic appearance or physical and intellectual performance of oneself. Another major cause for concern is germline will have effects on generations to come. Germ cells is the mutated alleles are normally counter-selected by evolution, which instead of using genetic engineering, would be artificially placed and fermented into the population. While this may help humanity as a whole, many may not want this evolutionary change that has been burdened on them, whether it is good or bad. This led to another interesting point on the topic, and is that of changing pathological condition compared to changing of a simple unfavorable characteristic or mental characteristics that are often indistinct. An example of this is dwarfism compared to a person's short stature. Then creating a movement that not only solves diseases and sickness, but also extends into creating a group of, quote, normal somatic characteristics across whole populations. Another key problem is how do we balance survival and this exploitation? A problem is where do we balance our priority? Should it be in editing genomes, or should it be in looking for healthier lifestyles? As the knowledge of molecular mechanisms develops, this creates the thought of moral, political, sociological, and religious debate. This goes back to the concern of cell manipulation and what is the ultimate purpose should be, whether it is the prevention of hereditary diseases, which is the most realistic and most needed use, or the ability to change embryos as wanted for reasons like cosmetic appearances or gene doping.